what I'm gonna tell you about is my experience with the use of machine learning and artificial intelligence. But first I want to give a plug that October is National Medical Librarians Month. And the reason this is important is that we all have to acknowledge that librarians have been the custodians of knowledge for quite some time. And even though we're all switching to digital, because the libraries are getting less and less space because the journals are electronic, there's still, I think, a specialty called medical librarian, which often gets neglected. So there's a plug about that. The obligatory disclosure. So I occasionally work with the pharmaceutical industry, and my spouse is a full-time employee at Genentech, which is a subsidiary of Roche. And I guess today we're going to talk about machine learning. So those of you who have already seen this part, just bear with me, but I think it's fair to say that humans can't even agree which year it is. And there's different ways to count it. My favorite one is the Mayan calendar, which ended in December 2012. Some of you might remember the books related to the topic and Fingerprint of the Gods and all those things, Graham Hancock. We were all going to be dead by now. I guess predictions can be wrong, right? But the problem with those predictions is that every prediction or even everything we do requires convention. The fact that I use words and you understand those words is because we agree that certain words carry certain meaning. And even in medicine, we need to navigate through concepts, so there's a time and of course space aspect of it. And in disease ontology as well, we need to take that into account. So as we make progress in data science, we need to develop solid relationships with conventions, and without that there would be not enough progress. But what bothers me sometimes at night is what do we know? So the very definition of knowledge, there are books written on the subject. Say I show you a picture of Avi, and I tell you that guy name is Tudor Oprea, and you will believe that he's Tudor Oprea until somebody says, no, wait, that's Avi Mayan. So when you knew he was Tudor, was that knowledge, right? So how do you define knowledge? How do you define truth? So what do you know and when do you know it? In science, that's not a trivial answer. One of my favorites, so I frequently go between Albuquerque, which started here, to Gothenburg, which is over there. And on airplanes, they show this map. And then on this projection, Greenland is huge. So this will challenge two aspects. One is the surface correction. Greenland is actually puny. Africa is huge. So go back, Africa, Greenland. Yeah. Look what the reality is. Yeah. And the other reality is that who says that Europe should be on top of the world? This, of course, was designed by Australians. And any Australian in the room? No. Okay. The point being, we live through conventions and we assume them to be true. And they rarely are true. So we just have to take that into account. Another one is that correlation is not the same as causation. So you could publish in Nature this type of correlation 30 years ago. Times have changed. But the short of it is, just because we find a really solid relationship between our data and what we believe to be the hypothesis doesn't mean that the hypothesis was true. So in principle, machine learning and AI are good at recognizing patterns. When I started in the field, the name was pattern recognition. It wasn't machine learning. Machine learning became catchy about 15 years ago. Now it's AI, but it's the same thing. It's still pattern recognition. And these models will not help you understand, but they will help you generate hypotheses. I guess part of the problem is that the biology doesn't apply, you cannot really apply a quantitative language. And that makes a reference to a paper by Yuri Lazebnik, which is how biology describes the radio. And you should look it up. It's a fascinating read. And of course, the other problem is we have confirmation bias. So when we have a hypothesis and we write an experiment and we get the results and the results confirm what we initially thought, then we assume that to be true. And that may or may not be true at the end. So in short, when you try to build machine learning models, you need to rely on different building blocks, and each building block has different values. So there's data, there's information, there's knowledge, and they do not overlap with each other. And of course, none of them relate to wisdom. I think wisdom relates more to failures and the ability to avoid failure as you develop your system. And I guess the other thing that we've tried to develop within the Knowledge Management Center that some of the people in the room are part of, we're really trying to understand how to build a knowledge repository for proteins. There's a distinction between artificial intelligence and machine learning. Machine learning is essentially building a model with a specific purpose. So you'll have an algorithm that will improve through experience, whereas artificial intelligence is replacing human capabilities with a computer. So think that in the 1980s, a pocket calculator was a precursor of AI because it was able to run complex mathematical operations that normally would require the human brain to do. So first, AI indexed mesh term, it dates from 1951 when Fletcher came up with the tortoise robot. Of course, this was a model, not a real robot. But since then, there's tens of thousands of publications that get indexed to that mesh term. And of course, we all recognize that AI is a moving target. My daughters interact with Alexa at home as if Alexa is a person. They talk to Alexa, they yell at Alexa, they interrupt Alexa, and to them there's no difference between talking to each other, so I have three girls, or talking to Alexa. So it's definitely an increasing number of companies that use AI in drug discovery, 158 startups as of October 2nd, and 33 pharma companies that use AI. And I will circulate my slides with Avi, so you will be able to check all the links that are in this. And of course, not everyone agrees that AI is the future of drug discovery. One of my favorite quotes comes from Amos Tversky. My colleagues, they study artificial intelligence. I study natural stupidity. I think, in my opinion, there is a lot of stupidity that goes onto the hype that generates the funding that goes into these companies. One example that is pretty much public knowledge is how IBM Watson Oncology, MD Anderson invested $60 million into that project. 3 years later, the head of MD Anderson was let go because there was nothing to show out of that program. So one of the take-home messages that we do live in an era of big data, and so data has been embiggened, and embiggen is a word that was first used by Lisa Simpson. It's now been adopted. It was one of the words of 2017. But how much of that data is true remains to be seen. And at least in my opinion, I do not think that AI can sift through data from false data. So a little bit about illuminating the druggable genome. We have quite a large project. There's lots of members of the consortia. Some of them are on the East Coast, some of them on the West Coast, and UNM is somewhere in the middle. And then there are the 6 which have dots. Those are the cutting-edge informatics technology centers. So there is a quite broad consortium. It also has components in Europe, so EBI and Novo Nordisk Foundation for Center for Protein Research. And then there's a team that works with KMC in Romania and another team that works with the KMC in India. So we pretty much cover quite a large space. As far as funding grants go, there are 3 experimental centers working specifically on the 3 druggable protein classes, G protein coupled receptors, ion channels, and kinases. There is the Resource Dissemination and Outreach Center, the Knowledge Management Center, of which part of is here at Mount Sinai, and then the Cutting Edge, which just started in April. And if you want to learn more about this, just go to druggablegenome.net. So part of my story with IDG starts with this analysis, which took about 7 years to make. Essentially, we spent 7 years mapping as much as possible all the drugs to all the targets. And that took us, comparatively speaking, we ended up comparing notes with 2 other centers in the UK. One was John Overington at DBI, and the other one was Bisan Al-Lazikani at the Institute for Cancer Research. Getting agreement on what's a drug target or not is not a trivial task. This paper pretty much covers all the FDA-approved drugs, so about 1,600 drugs and roughly 900 protein targets, of which about 600 are human. And the point of this graph is to illustrate that there is not a lot of innovation in the drug target space, in the sense that, temporally speaking, most of the drug targets have been discovered before 1990. So that's the blue line over there, so that before 1990, and if you look at this circular histogram and think of it as an eclipse, you'll squint and you'll see it has a blue halo. Most of the drug targets have been discovered before 1990. There's some new pockets of innovation. Cancer and kinases are over here. There's new drug targets in diabetes at about 1 o'clock, but not a lot of innovation. So with that in mind, we started to process information from across multiple sets of data. We're all about integrating large data sets, multiple resources, and once we process all the genome and gene-centric data, we realized we have to actually develop some of our own dictionaries. So we're working with the Monarch initiative to apply the Monarch disease ontology. We use Uberon. We also have some chemocentric databases, and essentially that's our playground. We go from literature to drug labels and clinical trials in patent space. And for patents, we use Shore-Campbell. So we fund the part of Shore-Campbell that does the annotation of proteins. They do automatically the annotation of chemicals. And once all that gets fed into Target Central, essentially we try to figure out how to deploy a variety of methods to visualize and catalog the data. And machine learning is one of the reasons I'm here today to discuss with Avi and his colleagues about it. Currently, version 6 of the Target Central database, you see how dynamic the set is. The ones that are highlighted in orange are completely new data sets. So the completely new data sets include the Cancer Cell Line Encyclopedia, as well as String for protein interactions, ClinVar, Links, which is another project some of you are familiar with, and the rat genome database. And then we, of course, have to update resources on a constant basis. And based on that, what we've developed or proposed is a knowledge-based classification for proteins. So the way we do that is we catalog information across all these channels, and those targets that we know to be drug targets are what we label clean, and they represent about 3% of the human proteome. Those that have chemical matter, which we have some potency criteria to allow it in that category, that's about 1,600 proteins. Those are what we label as TCAN. And then the way we label the understudied proteins, or T-dark, is essentially we have a fractional publication count, and we put those targets that have less than 5 publications into that bin. So let me show you a little bit where that comes from. So imagine that's the PubMed score that was first proposed by Lars Jensen, who is part of our initiative. So the way that works, imagine you have 100 publications from PubMed, and you're looking at the insulin receptor, and then there's one paper that talks about the insulin receptor, so then that paper gets a count of 1. But then you have 99 papers that talk about 20,000 proteins each. So then that adds 99 over 20,000 to the fractional count. So to get a fractional publication count of 5, you either have to have 5 dedicated papers or lots of very non-specific papers, and those are quite rare, in fact, to get to that point. So you need some specificity to get there. And the other thing we noticed is, so we catalog information from Antibodypedia. Of course, it's not possible to count antibodies by unique sequence because that's not disclosed. But what we try to do is at least count non-specific as well as monoclonal antibodies. And what we found is that roughly at 50 is the sweet spot where a protein is not enough of an interest, so it doesn't get lots of antibodies. So going back to TChem, this is how we use all the drugs that are approved by multiple target categories. So for kinases, for example, the specificity of that drug has to be at 30 nanomolar or better, whereas for ion channels, the gap is much lower. So it's roughly at 20 micromolar. And that's where the cutoff is for. So it's an individual cutoff for the target classes. This is the breakdown of the knowledge-based classification for the fractional publication count. So the median for T-dark is roughly at 0.3, and it jumps 100-fold at the next category. And then, of course, it jumps even higher for T-clin. And it's not just a relationship that one can observe with respect to publications, it's also what people are interested in. So one of the most accessed resources when it comes to protein interactions is the string database. And we asked String to give us the exact queries that were submitted to String over a 365-day period. This was in 2017. And it turns out that, so there's two ways you can go to String. Either you go to String directly and type a protein name, and you generate a query that way, or you go to Gene Cards or UniProt or some other resource, and you link out to String. And both of these illustrate that the pattern is the same. Dark proteins are just not subject of a lot of queries. We've expanded this analysis of dark versus T-clin targets to the set of genes that are relevant for rare diseases. This is a analysis that's just been accepted in Nature Drug Discovery, which I'm part of. So essentially what we've done here is to take all the known rare diseases and their disease associations to specific genes, and then ask the same question, are these genes in which category? And it turns out that, not surprisingly, a lot less of these genes are dark, and that's partly because once a protein is associated with a disease, it starts to get studied a lot. But the interesting part is that, for example, there's a lot more drug targets, so about 400 of them, that are associated with drugs. And in principle, what that means is that there could potentially be repurposing opportunities that could be at least considered for those rare diseases. Another resource that makes use quite frequently of what we do with the IDG is the International Mouse Phenotype Consortium. IMPC has 18 labs on 4 continents. Their annual budget is about $50 million, and their goal is to generate knockout mice for every single gene. And they're, however, interested in prioritizing the dark genes. And in fact, they're the only initiative that I'm aware of that deliberately hunts what we call T-dark. And we've worked with them for the last 4 years to help them prioritize genes. So I hope I illustrated convincingly that there is a knowledge deficit when it comes to proteins. So what we think of pharmacology and biochemistry, that pretty much represents about 11, even if I'm generous to say 15% of the proteome, that still means we have a lot of work to do. And there might be some opportunities in the space of rare diseases when it comes to the presence of TChem and TCLIN in relationship to those diseases. So while you watch this video, which is based on artificial soccer players from Brazil, those of you who know how Neymar has been playing would recognize that fall. I guess a lot of us have RoboCup 2018 was in Toronto, and I could have taken the GIF from that as well. It's not a lot of progress has been made. But the whole idea that AI is evil has been permeating mainstream. I don't think that's true to the point that I don't think there is a AI to speak of. So AI is a concept that, at least for the moment, doesn't exist. However, if you search for bad AI on Google Images, you'll find these two individuals associated with bad AI. And yeah, that might be the case. So part of the problem why AI is not there yet is we don't really know. We operate with concepts that for humans are easy to understand, but for computers, that's not really the case. And the reason for that is pretty much this, that to a computer, everything has to be black or white, true or false. And yes, there are multi-label classification systems, but most people use binary classification systems. And it turns out that for us, truth changes on a temporal basis. Truth evolves. In fact, we accept the fact that the TV weathermen tell us lies, but we watch TV weather all the time, right? So we're capable of distinguishing half-truths and half-lies, this whole alternative facts situation. And the other problem is that, to my knowledge, there is no mathematical model to discriminate truth from falsehood. And because of that, whatever you feed the AI for the machine learning system, if you feed it bad data, it will generate bad models. So that really needs to be understood. So that also applies to disease. What's a disease? Is it the absence of health? Is health the absence of disease? Those are circular definitions. So part of that problem is, how do you classify a disease? And this is text that I lifted from the abstract that Victor McCusick put in a paper in 1969. Nothing has changed. There is a tendency to classify diseases by gene, and there's a tendency to classify diseases by symptom. And both of them have valid arguments, but neither of them is absolutely correct. So you can't just pick and be in one camp or be in the other camp. And when it comes to how a computer classifies a disease, that still has not been solved or cannot be solved. So I just wanted to introduce you to the leaf concept in disease ontology, and this is a picture from the monarch ontology. Say someone is interested in congenital heart disease, that's not a leaf node. So what you have to recognize is that the disease could be long QT syndrome or the tetralogy of Fallot, etc. And then congenital heart disease itself is a heart disease or is a congenital anomaly of the cardiovascular system, etc. So how you count diseases is also a problem. What we're proposing in a commentary together with the people from the Monarch Initiative is to revise the number of rare diseases from 7,000 to over 10,000. They got very uncomfortable when they realized that this number has changed. So in an earlier version of the commentary, we had 10,500 plus. Now we're under 10,400, and they're like, okay, so let's just agree it's over 10,000. And I think, in general, I think that's a good point to make. But what we wanted to have as the title of the paper, which they said we cannot do because of style in nature drug discovery, is that if you do not count rare diseases properly, then rare disease patients do not count. And that's the end statement of the commentary. So what we're calling upon the WHO and other organizations is to figure out a better definition of rare diseases. Perhaps you're not aware of it, but the rare disease definition includes economic factors. The United States is the only country in the world that defines a rare disease by the ability of a pharmaceutical company to make profit. It's in the US law that defines the rare disease. I'm not joking. If a company cannot make a profit, then it's a rare disease. Think about it, how skewed our society is. And then, of course, the other definition is how many patients per X population. But the problem being, initially when the law was enacted in the 80s, it was 200 million Americans, now there's 300 plus million. So that number has to be revised almost on a constant basis. Therefore, should not surprise you, this is also a number that comes from the analysis that's coming out. There is a huge gap, because this is a log scale, between science and translation. So these are the number of orphan drugs that are specifically approved for orphan diseases by the FDA and the European Medicines Agency, which are pretty much in the 30s to 40s, whereas the number of papers exceeds hundreds of thousands of papers per year. And we're making progress, but it's very slow. Why that is? It's, for example, because we don't really know how to evaluate and prioritize drug targets in the drug industry. So a lot of publications have been done about protein X being associated with disease Y. Sometimes it's an overexpression or underexpression, and people write a paper and immediately they think they found a new drug target. The situation is a lot more complicated. The metaphor I like to use is the pig and the butcher. They're both involved in the process, but only the pig is committed. So what you really need to do is figure out which target you can commit clinically so that you can get a clinical effect once you hit it with a drug. Otherwise, nothing will happen. And of course, the other problem is we tend to split proteins at the gene level, and we forget that they form complexes. So if you search the literature for green2A or green1, of course, you will find that they have a certain function, but it's actually their combination in this glutamate and NMDA receptor, which is the drug target, because none of them alone would function as a drug target. Once you take all this into account, so what we've set to do is a protein knowledge graph, and think of this as the model of the underground, the subway system, and each of these are like stations, and then you can travel between different stations along different lines. And some of the lines will take you on the associations, some will take expression, some will take link signature, mode of action, all sorts of relationships can be established. And out of these, essentially what you try to capture is genotype, phenotype, interactions, pathways, structure, function, and then expression. And when you put all these things together, you can apply a mathematical model that's based on topology, so network topology, based on something called the METAPATH approach. And I don't particularly like the name METAPATH, but I didn't invent it, and it's kind of useful. So think of it as data and metadata. So a path would be that KIT regulates mast cell chemotaxis, and it's activated in asthma, whereas a different path would be that a kinase regulates mast cell something in a lung disease, or an even more general path would be that the gene regulates a process in a disease. So when you try to take this type of meta path into account, you can then start to ask the question, how can I link my protein with a specific disease? And when that link exists, we set that bit to 1, and we apply in the machine learning, we consider that to be a positive example. And when we find that the relationship is in general the same, except that link, the direct link doesn't exist, and we try to find it, we set that bit to 0. And that's how we generate positive and negative labels. And I am aware that we're still back to the question of what's true and what's false in the binary definition, but that's how classifiers work. So these are currently all the data that we feed the machine learning. We generally assume that everything that's in OMIM has a negative label when it comes to our search, unless it's specifically using data from OMIM. And this is just a visual implementation of that, so we actually have a lot of post-processing that goes on. So you start with constructing your database, and then you assume the assertions, and then you run the transformation into the metapath, and then finally you run the algorithms, and you get your results. So here's where it gets interesting. I spent time taking all the OMIM data from type 2 diabetes, trained a model, and got something that just looked on the left-hand side, got something that looked like this. So out of 51 genes, I had the curve 0.7, which is not terrible. I looked at the first descriptor that was identified by the model. It's hemoferritin. That one's mutated in hemochromatosis. 80 to 90% of people with hemochromatosis develop diabetes. So it kind of makes sense. You'll also see things like regulation of gluconeogenesis, autophagy. So there's a few things here that make sense, so I didn't think it was a terrible model. So I sent this to Mark McCarthy, who at the time was leading the type 2 diabetes consortium. And about a month later, he sent me a response saying, we don't really see anything in your predictions that makes sense according to our results. In fact, once we take a look at it, we question the training set. I had missed that this came out from OMIM, which is simply not a reliable source of information. Now remember, OMIM is 50 years of physician-curated literature. So it should be the state-of-the-art when it comes to medical data. And it probably has to be all revisited, or much of it. So we built a separate model. So I got from Mark and his colleagues, I got a different set of genes, which was 54 genes. And then the resulting model was completely different. In fact, if you compare these two, they're not very similar at all. Another model that we built was on autophagy. The way we set up to do this was essentially to recruit an expert in autophagy, Vojo Deretic. He was among the first 6 people in the world to work on autophagy. We started with a set of about 100 genes that we tried to build as positive labels, and then for negative labels we used OMIM, of course those that were not associated with autophagy in the first place. And we got a decent model. We put this on the internet, and then we submitted this to PLOS ONE. Both reviewers from PLOS ONE essentially complained that we did not have negative labels. So what they were saying is, we don't really think you have enough negative data, and how do we know that the models that you built are correct? So what we did was to revise the entire model, and essentially went to more recent literature, found a particular paper from Chris Schumacher, who had discovered a completely novel gene that was subject of a paper in PLOS ONE. But what he did in that paper was to say, we use these 5 experimental settings. So these are 5 sets of experiments that were carried out using CRISPR-Cas. And what I did out of those experiments was to pull out the supplementary results and extract 1,200 genes that all experiments agreed had no contribution in autophagy. And out of those 1,200, 200 plus matched the OMIM set, so we generated a model just on 100 plus versus 200 plus. And the two models are indeed very different. And what we've also done is to ask the question whether the links data set helps with the model or not. And we believe that links adds more noise than true data. And that's kind of our conclusion, and this is going to be the revised version of the paper. The good thing about this, so I'm going to retract it from +1 because the editor of autophagy, so this is impact factor 10 versus impact factor 5. The editor of autophagy saw our bioRxiv publication, liked it, and so we're going to submit autophagy. And it's probably going to end up in there soon. So the lesson I learned from this is that you always have to look at the results, try to challenge your assumptions, and then ask the question how to validate the model. So part of what I learned with machine learning models is that they all require what's called a ground truth. And this quote from Shakespeare, "The fault, dear Brutus, is not in our stars but in ourselves," that we are underlings. The fault is in our data. So if you do not have a good machine learning model, it's not because of the algorithm, it's because of the data. However, you do need data to build a machine learning model. So part of the reason I'm presenting all this learning experience, like we started with the model, we submitted it, and then we realized, okay, we need to revise it, generate a completely different model. The idea behind this, and the diabetes story as well, is that you need expert knowledge to help you validate the model. So unless I talk to the expert in type 2 diabetes or to the expert in autophagy, I don't even know if my models make any sense. So without that, it's all fluff. Or in other words, in God we trust, all else bring data. Yeah. So we really need that. And even this is controversial. Who said it first? It usually gets attributed to Deming. Turns out other people have been claimed to have said it. The only record I have is this guy, Bernard Fisher. He said it to a journalist in the early 1980s. So that was the first record of In God We Trust, all others. So one paper that made the waves in the last month is generative chemistry. Alex Javronkov and team from In Silico Medicine. They started day 0. By day 21, they submitted 8 molecule candidates for testing at WuXi, and 2 of those were in the nanomolar range as inhibitors for DDR1 kinase. Of course, this was presented in Forbes magazine, Wired, a number of Fortune, a number of magazines as drug discovery in 46 days, or 21 days even. So of course none of that is true. They didn't discover a drug, but they did discover new chemical property, so a new intellectual property. So I can confirm, because I was involved in checking this. I'm the author that didn't make it on the paper, so I'm in the acknowledgements for reasons that I'll be happy to tell you about. But the short of it is they are novel chemicals, and they have been designed by a machine. Okay, so they used TensorFlow, they used algorithms to come up with new chemical structures. I do think that in medicine, this is the next level of artificial intelligence. So Siemens have designed systems that are now capable to do perfectly contoured organs using deep learning. And the purpose of this is to provide the radiation oncologists with the exact location of where you're going to place your radiation therapy. And it's meant to improve treatment accuracy, of course, reduce the dosing, etc., etc. When it comes to natural language processing, so I was talking earlier to Gaurav about this. Tomasz Mikolov from Google came out with Word2Vec, where he was able to probably for the first time realize that a machine can actually come up with this type of solution. So king minus man plus woman equals, and it came out with queen as the answer. So it turns out that there is similarities between man and king and woman and queen, and you can partition words into little bits that each one gets assigned a number, and then you do vector analysis and you come up with this type of answer. And we even have a online Klingon translator. So those of you who are Star Trek fans, Klingon anyone sounds Tuhan Bai, okay? So that's really Klingon over there. But I think in medicine, we're not far from that either. I think it's possible to take all the data from patients in real time and then try to ask to come up with a platform that will try to take into advances in literature as well as biomarker data, omics, etc., that are specific to the patient, and then try to provide new practical answers. So there might be an Alexa Health. Alexa Health will know what my health status is. It will know what my calorie budget is, and you can ask Alexa, what should I shop today to make food? So those kind of things are coming. The technology is already there. It just hasn't happened yet. Also, a lot of you probably heard that Google is better than humans at this, Google is better than humans at that. Everyone these days has a way to beat radiologists at image processing. It turns out that they skewed the data. This came out in Lancet Digital Health, so they analyzed a lot of publications that made this type of claims, and it turns out they weren't entirely factual. This is what you would call alternative facts, okay? So whenever and even the headlines, so this paper came out and the headlines in The Guardian was, AI is as good as humans. But that's not what they said. What they said was, AI, the studies that favored AI gave the humans more difficult problems to answer. So there's a skewness in the data that wasn't properly reported, so they weren't asked the same questions, and the data were not deposited properly. So essentially poor reporting of the data. There's no why in AI. So you have to think of it this way, that artificial intelligence does not have curiosity like children do, and they're not very good at generating or acquiring new knowledge. And the reason, part of the reason is that people are part of the problem. In my opinion, just because Watson won Jeopardy doesn't mean Watson can discover drugs. In fact, Watson for Drug Discovery is being closed. So IBM had a program called WDD. It was around for about 5 years. It's being discontinued. Even though we think that in medical imaging AI is better or equivalent to humans, it turns out that those were skewed results. And that's partly people being part of the problem. So we have this whole alternative facts situation. And also, if you follow John Ioannidis, 75% of biomedical publications are false. So that's a statistic that makes me depressed. But if you look, so Amgen and Bayer published papers where they say we took data from high-impact journals and we were able to reproduce 1/3 in the case of Bayer and 11% in the case of Amgen. So science and nature publications tend to be less reliable than Journal of Biological Chemistry. So think about that. And then, of course, if the data you feed the AI is false, it's not going to help. So that's my last message. Truth is already here, it's just not evenly distributed. And this is our weakest point, the veracity. So what we trust and how we build our assumptions is going to limit our success. So with that, I'll stop.